So just a little bit of introduction of what this seminar series is. This is something that we're doing at OSU as part of our neurotrauma lecture series. To have one that kind of is outwardly facing. We're doing these little research in progress here at OSU, and people wanted to join in, and I wanted to have a platform so that OSU faculty could talk and reach a broader audience. And we're using these Buckeye Neurotrauma on Zoom to do that. And so today we have a former OSU trainee giving the lecture, Dr. Faith Brennan, who's now assistant professor in the Department of Biomedical Sciences at Queen's University in Ontario, Canada. So Faith did tremendous as both a graduate student and a postdoc. She was Wings of Life fellow here at OSU. She was a postdoc with Phil Popovich. Before that, she got her BS from Australia and the University of Queensland, where she also did her PhD with Mark Rutenberg, who I think many of us know, in the neurotrauma field. She's highly productive, 24 papers, 14 first author papers, and 10 co-author papers. And her connection to the chronic brain injury program is that she's had a chronic brain injury program pilot, and she also had the chronic brain injury paper of the year, which I think she'll talk a little bit in her data set today on microglia elimination and the effect of after spinal cord injury. And as a postdoc, she had a Craig Nielsen Fellowship and a Career Development Award from OSU. So really spectacular level of grant funding as a postdoc, and I'm sure that she'll continue that as a assistant professor there in Canada. So Faith, delighted to have you and see all the great work that you've been up to in the 7 months that you've been away from OSU, and look forward to seeing what you have to say. Thank you.
Thank you, Dr. Gobat, for that very kind introduction. I'm absolutely delighted today to share the Paper of the Year talk. It's a real privilege to be here speaking to everyone today about the work that I did while I was a postdoc at OSU. Before I begin, I'd like to state that I have no significant financial interest or relationship with commercial interests. This presentation will not include discussion of unapproved or off-label use of commercial products or devices. I'd also like to begin by thanking everybody who was involved in this work at OSU, especially everybody in Dr. Popovich's lab and Dr. Popovich, Chrissy Kegel, Jody Hall, and Zengan in particular. I'd also like to thank our collaborators at OSU Biomedical Informatics, particularly Dr. Chin Ma and his team. And of course, the many collaborators in the Department of Neuroscience, Dana McTighe, Jonathan Godbout, Andy Fisher, and their students and staff. Also, our collaborators at Harvard Medical School, Dr. Zigang He and Yin Li, and our sources of funding that made this work possible. So because this is a talk delivered to both a general and a specific audience, I thought I would overview some of the anatomy and histology of the spinal cord, since this is really going to come up a lot over this talk. So the spinal cord is essentially a highway of neural tissue that connects the brain to the rest of the body. So it's divided histologically into the gray matter and the white matter. The gray matter is the sort of central butterfly-shaped region that contains the cell bodies. You can see the cell bodies stained here in purple. This is surrounded by white matter, which contains the axonal tracts that go up and down the spinal cord. The spinal cord itself is positioned within the vertebral column. So on the ventral or dorsal side, we have the vertebral bodies. On the ventral or front side, we have the vertebral bodies, and on the dorsal or back side, we have the spinous processes of these bones. Now, sensory information comes in through the dorsal side of the spinal cord and sort of the tips of the butterfly wings. It's integrated in the central gray matter and up to the brain. And motor neurons relay motor information from the ventral horns down here. So in this way, we can get integration of sensory, motor, and autonomic information. We can have coordinated voluntary and involuntary actions in our day-to-day life. So what happens when we have a traumatic spinal cord injury or SCI? So this is a mechanical trauma that causes immediate cell death and axon disruption at the site of injury. And this is often because the bones, either the vertebral bodies or the dorsal spinous processes, can compress or contuse the spinal cord tissue. And this is associated with significant functional impairments in the motor, sensory, and autonomic system. And when we also see many complications, including metabolic syndrome, gut dysbiosis, cardiovascular dysfunction as well. But for this talk, we're going to really focus more on the motor deficit. So because the spinal cord motor neurons are in the ventral horns here, in the mouse, most of the motor information is relayed through the ventral and lateral white penetrates. So when we have a spinal cord injury that compresses down on the spinal cord, we can see there's a loss of function that correlates with the extent of the lesion. When we think about demographics and how many people are affected by spinal cord injury, there's around 17,000 cases every year in America. So that's around 1 every 2 hours. The median age of injury is around 43. And this has increased over time as we're seeing an aging population and an increased incidence of fall-related STIs. And when you take into account the amount of care, support, and assistive devices, loss of employment, and socioeconomic factors, these are no longer a death sentence, but they incur significant financial and human burden. So it's a really devastating disorder that we're sort of working on to try and find improvements, ways we can improve outcomes. So how we study spinal cord injury in the lab is by using a rodent model of spinal cord injury. So to do this, we perform what's called a contusion spinal cord injury. So here we can see we surgically can remove the dorsal spinous process from the spinal cord column to reveal the spinal cord. We can stabilize it with forceps. So we can see here the spinal cord is exposed and the bones are clamped with stabilizing forceps. The animal is then placed under an infinite horizon impactor device. So this is a computer-controlled device so that all the animals are delivered the same force and sort of tissue displacement of injury. And using this model, we can compare how, say, a genetic knockout or a drug intervention changes injury outcomes. And what several decades of research have shown using this model is that the primary mechanical injury that destroys those axons in cell bodies is not the end of the story. There's actually a second phase of injury, which is called secondary injury. And these are the molecular and cellular events that include ischemia, excitotoxicity, and inflammation. And these molecular events effectively worsen neurological outcomes by expanding the damaged tissue into healthy, intact tissue. So you end up with greater tissue loss and worse functional outcomes. So histologically, we can see that compared to the intact spinal cord, at 1 month after spinal cord injury, this architecture is lost, and we instead see a dense necrotic core. We can actually model this and map this secondary injury progression over time using in vivo magnetic resonance imaging. So this is an anesthetized mice, so we can perform respiratory gating using breathing monitoring and image the spinal cord of the mouse after injury. So we can see before spinal cord injury, we can detect the butterfly-shaped gray matter in black here, and this is surrounded by very bright white matter tracks in white here. At 2 hours after spinal cord injury, we can still distinguish this gray matter and white matter pretty clearly. So this really highlights that this injury is not the major driver of all of these tissue changes, but rather it's all these secondary injury events that are occurring gradually over time. So by 7 days, we can see that this distinction between the gray matter and white matter is lost. And by 30 days post-injury, we can see again this tissue architecture is lost and the cord is even atrophied a bit, and we see this really dense dark region in the central spinal cord. Now, because secondary injury is a gradual process, it is amenable to therapeutic intervention. So we can effectively explore what are the cellular and molecular mediators of secondary injury that we can then harness to improve recovery. And what several decades, again, of research before my time and even during it has shown is that there are several inflammatory cells that really dominate sites of spinal cord injury lesions. And one of the major ones is macrophages. So these are immune cells, and we can see here in the human spinal cord, these macrophages stained with CD68 really dominate most of the spinal cord, particularly in the central gray matter. And this response is conserved in the rodent, so in the mouse. We can see that the macrophages are found throughout the central gray matter, and even chronically after spinal cord injury, we get this real dense core of macrophages within the spinal cord. Now, this conserved and very robust response suggests that macrophages may be a really important cell type that we could harness therapeutically to improve recovery, since they're so prolific everywhere and this response is conserved. However, the role of macrophages in spinal cord injury is a little bit complex, and this is partly because macrophages are derived from two ontogenetically distinct sources, or from two different origins, effectively. So we have first the monocyte-derived macrophages. These are macrophages that are originating from monocytes that come in from the bloodstream. So in response to injury, these monocytes are mobilized from the bone marrow and the spleen, and they then circulate through the blood and come into the damaged spinal cord. Because the blood-spinal cord barrier is broken down, these cells can then come in. And they mostly localize to the central lesion core. And several studies have shown that if we deplete monocyte-derived macrophages, we get improved functional outcomes. So this suggests that monocyte-derived macrophages worsen injury recovery, potentially by limiting axon regeneration or causing excess tissue destruction through secondary injury processes. So here we can see in green some monocyte-derived macrophages surrounding some axons labeled in red. What's been less clear is what the role of the second macrophage population is, known as tissue-resident microglia. Now, tissue-resident microglia, they reside locally in the spinal cord throughout life. They're derived from the embryonic yolk sac, and they are exposed to all of the normal CNS antigens because they're parenchymal cells. However, their role has been much more difficult to understand because we haven't really had the tools until recently to manipulate them because they're a CNS resident cell type. So fortunately, we now do have these tools to kind of manipulate microglia and interrogate their role. And we hypothesized that these microglia, unlike monocyte-derived macrophages, would be beneficial to recovery. Because when you think about it from an evolutionary perspective, it doesn't really make sense to have an entire tissue-resident population that is harmful to that tissue. So we hypothesized that eliminating microglia using new techniques will reduce neuroprotection and worsen functional recovery after spinal cord injury. So how are we going to eliminate microglia? As I mentioned, we were able to use a relatively new drug called PLX5622. It's manufactured by Plexicon, and it is a CSF1R antagonist. And this antagonist specifically kills microglia, but leaves the monocyte-derived macrophage population intact. So we can feed mice with a chow that's laced with this drug, and we can start feeding it before injury and take them all the way out to 35 days post-injury and examine their behavior and their tissue, and whether depleting microglia actually changed spinal cord injury outcome. So here we have the two groups. The vehicle diet is in black and the Plexicon diet is in white. We performed the 75 kilodyne T9 contusion spinal cord injury using that impactor device. We also pulse chase mice with BRDU. So this is a compound that is able to label proliferating cells as they're proliferating. And we can collect the tissue at 35 days post-injury and compare whether they have differences in their white matter sparing and inflammation. So one of the first things we looked at was whether microglia indeed depleted in the mice that had Plexicon diet. So we can see here in the top row, stained with P2LI12, this is a microglial marker. Animals that were fed vehicle diet have microglia robustly staining around the lesion core. They're not actually inside that central lesion core where those monocyte-derived macrophages normally reside. However, in mice that had Plexicon, these microglia were almost completely eliminated. And this is quantified here, showing that microglia increased in the vehicle compared to animals with Plexicon diet. So having confirmed that microglia are indeed depleted in these mice, we wondered whether this actually affected functional recovery. So to test functional recovery in mice with spinal cord injury, we use what's called the BMS scale, or Basso-Mouse scale for locomotion. A 1 is slight ankle movement. A 3 is plantar placement of the paw. A 5 is frequent stepping. And 7 and above is looking at things like coordination and trunk stability. And the BMS subscore looks at more finer aspects of locomotion, including tail position and paw rotation. And what we found was that in animals that didn't have microglia present, there were significantly reduced BMS scores and BMS subscores, with them less frequently stepping and having significant impairments in trunk stability and coordination. Here's a clip of a mouse that's had a spinal cord injury and was fed vehicle chow. You can see it's able to step and bear weight pretty consistently. However, the animal that has no microglia is really unable to consistently step and struggles to maintain its trunk balance and coordination. So next, we looked into the tissue of these mice to see whether there are any differences in tissue sparing. So we can stain the tissue for iriochrome cyanide to look at the white matter and neurofilament to look at axons. And we found that there was significantly reduced myelin and axons in animals without microglia. We can also stack these sections together and make 3D renderings of the lesions. And when we do this, we find that mice without microglia have significantly larger lesion volumes. When we looked at the inflammatory response at 35 days post-injury, we found that mice without microglia had a significantly more aggressive monocyte-derived macrophage response. These monocyte-derived macrophages were no longer confined to a central lesion core, but rather they spread throughout the ventrolateral white matter. So we see these big amoeboid-shaped rings of F480 positive macrophages, especially coming down and expanding throughout the white matter. We can also stain these with myelin basic protein, and we see that there is a significant increase in the size of these cells and a reduction in myelin basic protein in these ventral tracts. Now, why would there be an exacerbated monocyte-derived macrophage response when microglia aren't present? Well, one of the other resident cells in the spinal cord is called astrocytes. And these astrocytes talk to microglia, and they are really important for forming a border between the intact spinal tissue and spared spinal tissue after spinal cord injury. So these astrocytes can be labeled with GFAP. So we can see in the vehicle, they form a really strong border around this F480 positive macrophages. However, without microglia, these astrocytes fail to form a really robust border. Instead, we get this very fragmented loci phenotype, and we get puncturing through to the ventral white matter of these monocyte-derived macrophages. So this can be quantified here, showing the GFAP positive area along the length of the spinal cord is increased in the vehicle compared to mice without microglia. We also found that there was a significant reduction of proliferating astrocytes. So using that BRDU label to co-label with GFAP, we can count the number of proliferating astrocytes. And this was significantly reduced when microglia are depleted, rostral and caudal from the lesion. Now, we wondered whether is this impairment in the inflammatory dynamics and the astroglial border formation occurring early after spinal cord injury when microglia are depleted. So we generated new cohorts of mice and collected tissue at 7 days, 14 days, and 21 days post-injury. And what we found was that at 7 days post-injury, as has been reported by many groups previously, there's a very robust macrophage response. And this is sort of consistent to 14 days post-injury. And by 21 days post-injury, these macrophages are mostly confined within a lesion core and surrounded by a very robust GFAP positive lesion border. Now, the recruitment of monocyte-derived macrophages was significantly delayed when microglia are depleted. So you can see there's very few cyans here. And when these cells do come in, they actually come into sort of a different area. They localize more outwards in the spinal cord. And by 21 days, we see this phenotype very similar to 35 days post-injury, where these macrophages have expanded all the way out into the ventrolateral white matter, these regions that are really important for motor recovery. And concurrent with this, we see an impairment in the GFAP border formation around the lesion. And we could actually appreciate this impairment in lesion closure macroscopically when we dissected out the spinal cords. We found that compared to vehicle mice, animals with plexicon had significantly more sort of hemorrhaging pathology at the site that they were injured compared to controls. We next wondered if this phenotype was specific to the contusion spinal cord injury or whether it was consistent in a more severe crush model of spinal cord injury. So we chose the lumbar spinal cord, which has a very high number of astrocytes, and interrogated whether microglia depletion effects on lesion pathology could be replicated in this model. This is effectively the forceps that completely crushing the spinal cord laterally. Again, we were able to deplete microglia, and in this model, mice fed vehicle diet recover slight ankle movement after their crush, but mice fed plexicon and have no microglia have almost no functional recovery, even 2 weeks post-injury. We again saw this impairment in astroglial border formation around the lesion. We again saw reduced myelin preservation and expanded F480 positive macrophage presence outside that main lesion core. And we also saw, using grayscale analysis, a reduction in astrocyte border intensity. We also saw reduced numbers of neuronal profiles in this model away from the lesion. And using track tracing studies where we can inject an anterior grade tracer into the brain to label corticospinal tract, we found that there was increased axon dieback in this model in mice fed plexicon without microglia. And this is again showing this image from the start, where we can see that without microglia, these macrophages actually migrate out to these degenerating axons. And it appears that they're sort of more associated with blebbing-like axons compared to the control. So to sum up this first part of the talk, microglia are really important for recovery of motor function after SCI. And spontaneous endogenous repair processes that occur are really governed by microglia in some way. This includes formation of the astrocyte border and preventing monocyte-derived macrophage pathology. And this phenotype is consistent across both a contusion and a crush model of spinal cord injury. But this kind of begs the question, we have this very striking phenotype, but we didn't really understand the molecular mechanism for how this was occurring. So in part 2 of this talk, I'm going to explain more of our single cell and bulk RNA sequencing data. So what we were able to do was to perform SCI in animals that were fed vehicle or plexicon diet and collect out spinal cords at 7 days post-injury. So when we compare the transcription to the transcriptional sort of landscape of these spinal cords, we can understand what genes are increased by spinal cord injury and what genes are controlled by microglia. When we compare spinal cord injury to sham in the control condition, we see that spinal cord injury triggers a massive upregulation of genes. Many of these are immune response genes, including things like CD68, CD22, and several other factors. And when we compare animals with spinal cord injury that were fed microglia depleting diet versus control, we see that plexicon significantly reduces the number of genes in the spinal cord. And many of these are microglia linked or immune genes, including CSF1R, CH3CR1, and B2RY12. Now, if we look at the genes that are increased by spinal cord injury and are not increased when microglia are depleted, we see around half of these genes are dependent on microglia, suggesting that microglia control effectively half of the gene transcription that occurs in the spinal cord after an injury. And we can perform pathway analysis on all of these microglia-dependent genes. And I just wanted to show a summary table here showing all of the many different pathways that were identified. Several of these were involved in innate immunity, including toll-like receptor cascades, cytokine signaling, also adaptive immunity, metabolism, cell cycle, and hemostasis as well. So this hemostasis could actually potentially explain why we saw that striking hemorrhaging phenotype as well. Now, we really wanted to hone in on some specific mechanisms, and since immune factors came up so consistently, we pulled out the genes that were specifically involved in some of the top biological processes. So over here, we can see that the top genes involved in phagocytosis and cytokine production are shown here. You can see this big increase in spinal cord injury compared to the sham groups, and the reduction when plexicon was on board. And we can actually confirm whether this phenotype of gene transcription is consistent in the tissue by staining for phagocytosed lipid. So typically, cells that can phagocytose lipid can prevent it from being growth inhibitory or prevent it sort of leaving debris in the spinal cord. So consistent with our gene transcription data, animals without microglia had significantly reduced phagocytosis of lipids in the spinal cord. That is really important to be cleared for functional recovery. We honed in on a network analysis of sort of key genes that kept coming up in the key pathways that were involved in microglia-dependent injury response. And I'm just going to highlight two here that we identified in the paper that were also shown by several other groups to be really important for controlling, coordinating immune responses and functional recovery to spinal cord injury. And two of these genes were CCL2 and TLR2. And CCL2 is really important for coordinating monocyte-derived macrophage chemotaxis and their recruitment to the correct place in the spinal cord. And TLR2 is really important for expectively polarizing macrophages to a protective phenotype. So we wondered, could we actually potentially rescue the microglia depletion phenotype by reinstating these signals into the spinal cord and reprogramming monocyte-derived macrophages? So to test this hypothesis, we fed mice Plexicon diet and we injected them into the spinal cord early after injury with either TLR2 agonist and recombinant CCL2 or control injections. We also compared these mice to animals that did not have microglia depletion and that were fed vehicle diet. And when we do this, we find that, again, animals fed Plexicon with nothing else on board had significantly worse motor recovery compared to mice that were fed vehicle. However, mice that were injected with the TLR2 agonist and recombinant CCL2 actually almost completely recovered to the baseline levels when microglia were not depleted, suggesting that we can partially functionally rescue this phenotype. So what did the tissue look like? When we now analyzed the different groups, we found that mice with microglia depletion again had significantly poorer lesion pathology. This included reduced third myelin. But the mice that had the TLR2 and CCL2 reinstated were actually able to have improved tissue sparing. We were actually also able to rescue the astrocyte border formation and reduce monocyte-derived macrophage pathology. And we're also able to improve lipid phagocytosis in that central gray matter. Given that we found that some of these signals are really important for functional recovery, we wanted to dive a little bit deeper into the specific cell types that were coordinating recovery after spinal cord injury. So bulk sequencing is good because it gives us a panel of genes that we can then maybe manipulate, but it doesn't tell us what cell types are controlling those genes. So to better understand this, we performed single-cell RNA sequencing of control and microglia-depleted spinal cords. So we again fed mice vehicle or Plexicon diet and collected spinal cords at 7 or 28 days post-injury or from sham controls. We can then dissociate them to a single-cell suspension and capture and barcode them. So here we can see several different cell types that were able to be isolated from the spinal cord. And I'm going to focus on the microglia, the monocyte-derived macrophages, and the astrocyte populations, and how their genes responded to spinal cord injury and how they were affected when microglia were depleted. So we'll focus on the microglia first, and obviously this is just in the vehicle control and looking at how these sort of recover or change their transcriptional profile over time. What we noticed was that there were several different populations of microglia based on the distinct gene signatures. These included homeostatic, interferon producing, proliferating, lipid processing, and iron processing. And if we split out these different populations by time point, we can see very clearly that in the control, so the uninjured spinal cord, they're mostly dominated by homeostatic microglia populations. However, if we look at 7 days post-injury, we see that the microglia are predominantly proliferating cells. And at 28 days post-injury, the phenotype shifts again to be proliferating as well as patrolling and lipid processing. We also see a larger cluster of iron processing microglia, which could potentially be involved again in hemostasis. When we looked at the actual genes that were increased in spinal cord injury at 7 days versus sham or at 28 days versus sham, We see several genes that are consistently increased or decreased at both 7 and 28 days compared to sham. So in microglia, we see increased transcription of genes related to translation and metabolism. We also see iron homeostasis and pathogen phagocytosis pathways. So again, suggesting that microglia may coordinate these immune responses by proliferating and increasing genes associated with the immune response. Interestingly, they also decrease genes associated with B cell activation and myeloid cell differentiation and several other pathways, including apoptosis. Next, we looked at the monocyte-derived macrophage cluster to see how this changed after spinal cord injury and how it was affected by microglia depletion. In the sham control, these monocyte-derived macrophages are mainly patrolling cells, potentially that line the vasculature or circulating through. At 7 days post-injury, we see an increase in the number of lipid binding, calcium binding, and border-recruited macrophages, as well as antigen binding macrophages. By 28 days post-injury, we see an increased number of cholesterol processing and matrix remodeling and cathepsin inhibiting macrophages. Now, breakdown of the extracellular matrix is thought to contribute to expansion of the lesion core. And what we noticed was that in the microglia depleted animals, there was an expansion of these matrix remodeling macrophages and a reduction of cathepsin inhibiting macrophages, which prevent that breakdown. So this could potentially explain why we saw a sort of more aggressive phenotype of macrophages in animals without microglia, given that they're expressing more genes related to tissue breakdown and immune responses. Finally, we looked at the astrocyte cluster in the single-cell RNA sequencing dataset. And when we did this, we found that there were 3 major subsets of astrocytes. And we loosely sort of term them as transporter, metabolism, and inflammation based on the predominant genes that these clusters expressed. So for example, the inflammatory subset in orange over here expresses some complement receptors, Fc gamma receptor, whereas the transporter cluster here expresses transporter genes including SLC1A2 and ATP transporters. Now, if we look in the vehicle control mice, these clusters split over time, we can see that mostly the transporter and metabolism clusters dominate in the sham. However, at 7 and 28 days post-injury, the number of inflammatory astrocytes increases to around 40%. Now, this shift to inflammatory astrocytes fails to occur when animals have microglia depletion. And we can perform pathway analysis to see effectively what genes are increased in astrocytes and depend on microglia to increase. And these included things like translation and metabolism, several nuclear processes, as well as activity of the ribosome. And this could potentially explain why we saw pretty big deficits in astrocyte responses to spinal cord injury, given that these cells are not proliferating as robustly as they would in the control. Finally, we used CellChat to understand how microglia coordinate cell interactions in spinal cord injury. So in this sort of tool, we're able to computationally isolate our microglia, monocyte-derived macrophage, and astrocyte clusters, and look at all of the ligands and the receptors that they express. And we can see that we can essentially map pathways from known interactions onto the genes that are increased. So in dark green, we can see the interactions between these cells. So for example, here we can see microglia expressing PSAP or prostaglandin, and this signals to astrocyte GPR37L1. And this pathway is known to limit oxidative stress in astrocytes and promote their survival and proliferation. We can also see several interactions between immune receptors in microglia and monocyte-derived macrophages. And we can make these sort of circus plots for each different condition, including sham and spinal cord injury, and effectively condense it down into a diagram of what cell-cell interactions are controlled by microglia. So here we can see the sham condition, 7 days post-injury and 28 days post-injury. And in brown, we can see all of the microglial genes. In orange are the astrocyte genes, and in blue are the monocyte-derived macrophage genes. So what we found here is that microglial proserpinin signals to astrocyte GPR37L1 to promote astrocyte migration and survival. Microglia also signal to monocyte-derived macrophages to promote their adhesion and stabilization. They also signal through galectins, osteopontins, to integrins on monocyte-derived macrophages. And all of these ligands have been associated with tissue regeneration and tissue sparing, inhibition of B-cell activation, and regulation of phagocytosis. So collectively, this suggests that microglia are signaling to monocyte-derived macrophages and astrocytes through several different types of cell ligand receptors, which could potentially be exploited therapeutically to improve recovery. So to conclude part 2 of the talk, microglia are essentially important for programming the monocyte-derived macrophage response in a manner that includes CCL2 and TLR2 dependent mechanisms. They prevent dominance of monocyte-derived macrophages with a neurotoxic phenotype by several different cell ligand interactions. And they also interact with astrocytes to drive genes that promote their survival, proliferation, and glial border formation. So with that, I would like to thank again everybody who's been involved in this work and making this paper possible, especially Dr. Popovich for his support and mentorship throughout this whole project, Dr. Ma, our collaborators, and our funding. And I would also like to make a note that our lab in Canada is recruiting. So if you are interested or you know someone who's interested in doing graduate, postdoctoral, or technician position, please reach out to me by email or check out our website. And thank you again for your attention, and I'd be happy to take any questions that you have.
Thank you, Faith. Yeah, so we can do questions a couple different ways. I think we can have Q&A. If you want to raise your hand, we can have you ask live, or you can chat, put something in the chat, and we can read it off for you. Either way works. And Kedar has that in the chat too. We have a question from Phil.
Hi, Faith. So, great talk. You did a great job as always. I'm wondering if you can talk to us a little bit about, you and I, we talked a little bit about trying to interpret the single cell data and some of the bulk seq data, but there's a lot of data there, right? And so if you were to move forward, what microglial dependent processes do you think people should think about prioritizing as primarily reparative? And what would be some potential ways that you would think could those pathways or cell-cell interactions could be manipulated?
Yeah, that's a great question. I think one of the first ones that sort of jumped out to me was the proserpinin pathway. Proserpinin has been sort of indicated in some chronic neurodegenerative diseases in the brain, but it hasn't really been looked at as much in spinal cord injury. So I think what we would love to do is make a GPR37L1 astrocyte-specific knockout, and maybe also a microglia-specific proserpinin knockout, and see whether that affects any of the astrocyte responses or the microglia-dependent responses. I think that would be probably the main ones that I'd go after. There was also an array of integrins that are regulated on monocyte-derived macrophages by microglia, and although integrins are important for recruitment of immune cells and have been shown by many people, all the different sort of subtypes haven't been fully characterized, over time, and on which macrophages they're expressed by. So I think it would be also good to dissect out a little bit more which one of all of these integrin pathways is beneficial versus detrimental. So those would probably be the two ones I'd go for.
Thanks, Faith.
Yeah, I think that's the cool part about doing some of these microglia elimination studies is I think, and then doing the single cell, it kind of gives you potential targets to then kind of fine tune the pathways, because obviously no one's going to want to remove microglia, or that big of a change. So you use that to kind of fine tune. You know, my
Yeah.
question is, you know, with our TBI work, and we do diffuse injury in the brain, like when you take away microglia, there's tons of benefits long term. And yours, it's the exact opposite. So what is it about spinal cord injury versus brain injury why that would be the case?
Yeah, that's a really important question. So I think part of it is that it comes back to the anatomy, and it's different tracks and pathways of different white matter versus gray matter ratio that's being affected. Different density of astrocytes, I think, has a huge role. Effectively, you see a massive astrocyte border around spinal cord injury lesions that is not really as robust in the brain injury model. And I think also, expression of, different vascular endothelial molecules in the brain versus the spinal cord can control permeability of these organs. There's also, the brain has the circumventricular organs, and there's a lot of work on that sort of system and recruitment of immune cells from that sort of avenue. So I think it's also another factor that I think is important to point out is most spinal cord injury literature uses females, and most traumatic brain injury literature has historically used males. So I think because microglia are sexually dimorphic cells, and in our study we only used females, I think sex differences are also playing a role. And it would be really interesting if we could replicate our study in males and females. Mm-hmm. So I think, it's an anatomy and demographics as well.
Yeah, I think even in TBI, some of the penetrating injuries have a little bit different response to the microglia. If you have penetrating injury, really recruitment of a lot of peripheral immune cells, it's a different type of injury than if you have in the diffuse injury. We don't see that level of recruitment of immune cells. Got a question from Damon, which I can read off for you. It says, "Hi Faith, great talk. Follow up from Phil. Do you have any insight on timing or pseudotime for the microglial response? Do you know yet if there would be an ideal day post-injury to intervene on microglia function?"
So that's a great question. Hi, Damon. I think we know that if we delay microglia depletion even 1 week, we still see deficits. So I think, within the first 2 weeks, microglia are really important for functional recovery. Whether, what they're doing if we deplete them chronically and how they're affecting, more distant pathways in the spinal cord, maybe synaptogenesis in the lumbar tracts in this model isn't really clear. But I think I would probably say within the first 2 weeks, I think that's the most important time.
Great. Let's see. Do you have any more questions for Faith? Anybody from the audience? Any hands up? Okay, none that we see here. Oh wait, we do have one hand up.
Yeah, hi, I just raised my hand right now. Can you guys hear me?
Yes, thank you.
Yeah, great talk, Faith. I just had a question about this inhibitor, this chow. So how confident are you that it's specific for microglia? Because I've seen papers where they said, for example, so I work on TBI, so this same inhibitor also affects border-associated macrophages in the brain. So for the spinal cord, at least, or for your purposes, how confident are you that you're only wiping out microglia, not other cell populations?
Yeah, that's a great question. Border-associated macrophages, I think we may be wiping them out, but for monocyte-derived macrophages, we're not wiping them out. In our hands, we don't see any depletion of monocyte-derived macrophages. And this is consistent with other groups showing that the CSF1R is expressed by both monocytes and microglia, but it's not required for monocyte survival the way it is for microglia survival. So we did a big study where we gave mice endotoxin or LPS or control, and we looked at how their bone marrow, spleens, blood immune responses were. And we really didn't see any differences in cell proliferation, cell mobilization, or cell numbers. across these different organs and with our lexicon chow. So the border-associated macrophages, though, are a little, I would expect that they're depleted just because we didn't really see, the sort of what you'd expect labeling for them in when we did our histology. But definitely, I don't think we're depleting the monosaccharide macrophages in our hands using this dose of drug that we're using.
Awesome, thank you. Is that work published that you just said about where you did the different organs, or is that not yet published?
Yeah, it's supplemental figure 1 of this.
Okay, yeah, I'm sorry, I didn't read the paper,
Yeah.
but I'll go through it
That's
for sure.
okay. No, it's a long monster. So
Okay,
it's in the supplemental figures. Yeah.
awesome, thank you.
Okay. Let's see, any more in the chat, Q&A? Okay, I'm not seeing anything. So I guess Faith will get you off the hook here. But
Alrighty, thank you.
thanks again for joining us from Ontario. Is it the same time there? What time is it?
It is 10, just after 10:15.
Oh, same time. All right.
Yeah.
So you can get back to work on a Friday, and everybody could enjoy the beautiful weather here in Columbus. Those of you that are here in Columbus.
Yes.
And
Yes. Enjoy the weekend, everybody. Have a great day.
yep. Thanks again, Faith. Take care.